We are going to start this third session of the masterclass season. And we start the session by a presentation by Sébastien Déjean about data science in interdisciplinary research projects.
Okay, so thank you. So I hope that the sound is good for everyone and that my French accent is okay for everyone also. And a French accent from southwest France. Okay, maybe you can listen to it, but... indeed, I'm going to talk about data science in interdisciplinary research project. So first, I'm going to introduce who am I, just to show you that I am a kind of outlier in the mathematics department. So first, so here is a short resume. So I'm not so young now because I had my A-levels in mathematics and physics in 1993. Then I go through lectures in mathematics mainly. So I had a Bachelor of Science in industrial mathematics. Then a master degree in applied mathematics, nearly the same. We can find behind industrial mathematics or applied mathematics, depending on the lectures. After I get a PhD in applied mathematics and specialized in statistics for works concerning comparison of estimation methods for nonlinear mixed effect models and application to the modeling of the evolution of the leaf area index of crops observed by remote sensing. Okay, it's even longer in French. You can imagine that it can be long. After I was hired here as a research engineer in statistics at the university in the mathematics department, and I have also obtained my habilitation thesis in 2019. So in the mathematics department here we are more than 200 people, and we are 4 research engineers either in statistics or in high computing performance. So you can wonder, what does a research engineer in statistics actually do? So officially, you can find in French information on the website of the Ministry of Research. But in few words, a research engineer in statistics can be an engineer responsible for statistical aspects in a research laboratory. I would rather say, in my case, that I am an engineer responsible for statistical aspects in interdisciplinary research projects. I can manage statistical projects. I have to sometimes to define data collection and management and the associated processing chain. I can participate in national and international research projects and associated publications, and so on. In practice, I do like to wrap up everything in, okay, I try to contribute to answer questions in the real world. That's quite ambitious, but let's try to convince you that I'm trying to answer questions in the real world. So, what is a real world problem? So I do like to quote this book from Jordan Ellenberg. So, How Not to Be Wrong, the Hidden Math of Everyday Life. I can give you an advice. You can read it. It's very, very nice, very nicely written, so it's very good. And what is written somewhere is that, okay, the author blame word problems. So for me, word and world would be nearly the same. I'll try to focus on not on my pronunciation, but maybe on the text. So I blame word problems. They give a badly wrong impression of the relation between mathematics and reality. For instance, Bobby has 300 marbles and gives 30% of them to Jenny. He gives half as many to Jimmy as he gave to Jenny. How many does he have left? Okay, so that looks like it's about the real world, but it's just an arithmetic problem in a not very convincing disguise. So let's go on. But real world questions are not like word problems. A real world problem is something like, has the recession and its aftermath been especially bad for women in the workforce? And if so, to what extent is it the result of Obama administration policies? It's a real world problem, okay? But what is written after, your calculator doesn't have a button for this. Sure, because in order to give a sensible answer, you need to know more than just numbers. It's only after you have started to formulate these questions that you take out the calculator. But at that point, the real mental work is already finished. Whoa. Dividing one number by another is mere computation. Figuring out what you should divide by what is mathematics. Okay? And another quote from this paper, very nice to read, The Future of Data Analysis, but written in 1961. So we can see if what is written here to describe the future of data analysis actually occurred in the past few years. So the future of data analysis can involve grid progress maybe. I think progress. The overcoming of real difficulties and the provision of a grid service to all fields of science and technology. With it, that remains to us, to our willingness to take up the rocky road of real problem, once again, in preference to the smooth road of unreal assumptions, arbitrary criteria, and abstract results without real attachments. So I'm sorry for my colleagues in fundamental mathematics that are in the world of, in the smooth world of unreal assumptions, arbitrary criteria, and abstract results, but okay. My job is more to the rocky road of real problems. Maybe we could have a kind of survey. So, do you prefer a rocky road of real problems, or a smooth road of unreal assumptions, arbitrary criteria, and so on? Maybe we can have a survey. Who prefer this place? Yeah. Okay. Okay. If I'm being honest, I have to acknowledge that when I want to have a run or hiking in the mountains, before I have to take this road. So I'm not trying to oppose everything. Okay, so just, but I'd rather be there than there. Okay. Okay, so now let's talk about a real world problem. How is global warming affecting plant growth? Do you agree it's a real world problem? Okay. So I'm going to try to show you how, as a mathematician, as a research engineer in statistics or data science, I can help, I can contribute to answer this kind of question. But first, back to linear algebra. So once again, a code, sorry, it's a lot of text, but I read it for you also, so take it easy. So the biological science are today, so today it's in the 60s, okay, the past century. The biological science are today in the process of changing from being primarily descriptive to being very much quantitative. As a result, biologists find themselves confronted more and more with large amounts of numerical data. But the mere collecting and recording of data achieve nothing. Having been collected, they must be investigated to see what information may be contained concerning the biological problem at hand. Frequently, however, biologists have to subject their data to more complex calculations, requiring procedures that involve mathematical details beyond their general experience. In order to carry out the mathematics, the biologist in this situation must either learn the procedures himself or herself, of course, or at least learn something of the language of mathematics, that he may communicate satisfactorily with the mathematicians whose aid he enlists. Okay, so you see the link between, okay, I have a problem in life science, biological science, with global warming affect plants or not, and probably there are links to be done with linear algebra. So let's have a look briefly on mathematics. It's not what I have to say, it's not what I prefer, but let's start to something that you have probably heard about, eigen decomposition of a matrix. Is it okay? Have you already heard about that? Okay. So it seems that you can decompose a square matrix with the specific properties in this way. So V lambda, V transpose. Okay? So the columns RV are the eigenvectors of the matrix M, corresponding to the eigenvalues you have on the diagonal of the lambda matrix. And you have the definition, lambda is an eigenvalue of M if and only if it exists one vector V of length n such that MV equals lambda V. Okay? Maybe you have also heard about singular value decomposition, SVD for the friends. So in this decomposition, we have a rectangular matrix, M times N, big M, that can be decomposed in U sigma V transpose. So here we have left and right singular vectors corresponding to singular values we can find on the diagonal of the matrix sigma. And here, sigma is a singular value of M if and only if there exist two vectors, U length M and V length N, such that MV equals sigma U and M transpose U equals sigma V. Okay? So we are in the world of matrix algebra. So it can be a very easy exercise to see the link between the two decompositions. So if you start with decomposition by singular value decomposition of M, and you compute M transpose M. So you just use, here you replace M with SVD, here. You use this property, AB transpose equals B transpose A transpose. You use this property of vector U and you get this for M transpose M. In the same way, you can have M transpose with this expression. And so you can see that we obtain the eigen decomposition of M transpose M and M transpose with eigenvalues equal to the square of the singular values and eigenvectors respectively equal to left and right singular vectors. Okay? It's just a game between the two decomposition. So now how this matrix algebra is used in the role of statistics and data analysis or data science. So here in statistics, when you have a data set full of numbers, the first tool to use is principal component analysis. Do you know this method? Okay, so let's have a look at what is principal component analysis. And frequently I suggest to students in statistics, first PCA, then after you think. Okay, so the first step of data analysis, PCA. So principal component analysis. You apply this on a N times P data matrix. So PCA is an orthogonal linear transformation that projects the data in a new coordinate system such that the greatest variance of the data lies on the first coordinate. First PC, first principal component. The second greatest variance on the second PC, and so on. It can be shown, it can be an exercise, not too difficult, that the greatest variance is the first eigenvalue of X transpose X, and the transformation of the coordinates from the initial space to the principal component space is done using the first eigenvector, and so on for the other eigenvalues and eigenvectors. So just to try to link once again between matrix algebra and statistics, I've just this quote from Wikipedia. It was nicely written, so I take it like that. And maybe at the end of the long text, after you will have pictures and photographs and so on, so it will be easier to follow. So the singular values in sigma are the square roots of the eigenvalues of the matrix X transpose X. Okay, we have just seen this before. Each eigenvalue is proportional to the portion of the variance. Not exactly the variance, but the sum of the square distance of the point from their multidimensional mean. So, proportional to the portion of the variance that is associated with each eigenvector. The sum of all the eigenvalues is equal to the sum of the square distance of the point for their multidimensional mean. Okay, we'll see what is behind this later. So PCA essentially rotates the set of points around their mean in order to align with the principal components. This moves as much of the variance as possible. Okay, using an orthogonal transformation. And later, so the values in the remaining dimension, therefore, tend to be small and may be dropped with minimal loss of information. So PCA is often used in this manner for dimensionality reduction. And PCA has the distinction of being the optimal orthogonal transformation for keeping the subspace that has the largest variance as defined above. Maybe a little complicated to figure out what is behind PCA. So let's try another way to explain what is PCA. So I have a question. Would you use a cubic box to pack a fishing rod? Maybe for Christmas you would like to offer a fishing rod to someone. How would you pack this? In a cubic box? If you want to make a joke, okay, why not? But it seems it will not be so efficient. Okay, you order your fishing rod on Amazon or whatever. I'm not sure you will receive something like that. More probably a tube. Okay? So I'm still talking about PCA, okay? I'm not lost. So let's have a look at this now. Let's imagine we have people. I have measured shoulder girths, waist girths, and chest girths. Okay? So hello, I take the measurements and I have this kind of points. Probably I will not have so much people in this corner or in the corner there. Do you agree? We could say that we'll have people like that here with a small shoulder, waist, and chest girths, and on the other corner, maybe this kind of guys, okay? So, do we need 3 dimensions to represent what can be called standard individuals? Okay? Not sure. And it's the same question as, do we need a cubic box to pack a fishing rod? Okay? It's the same idea. So what could we do with this data in 3 dimensions? What could I do with it? I have seen something like that. Okay, I appreciate this. Because the idea would be to, okay, what if I projected my points on this one-dimensional space? Okay, so I have here my points projected there. So I don't have any more neither shoulder girths nor waist girths and chest girths. I have a synthetic variable, a new one, that I have built with my 3 variables, and I can interpret this as a kind of beefiness. Okay, beefy is tough. Okay. So on the left, I have the less beefy guys, and on the other one, the toughest like that. Okay. And here, if you hadn't run any PCA before, so I'm happy to tell you that we have just done a PCA. principal component analysis. So let's have a look at another toy example. So I have completed. I have 20 individuals or observations. I have 5 variables, so shoulder girth, chest girth, waist girth, weight and height. So here are the data. 20 rows, 5 columns. It's not a big data, okay? But if I ask you, okay, could you please tell me what kind of body shape I have in this data set? Are you able in, I would say, 1 or 2 minutes to tell me, okay, there was a cluster of this kind of body shape and an outlier there and so on? Difficult, okay? You would need time to focus on these numbers. So let's try to see what PCA can do for us. So here again are the raw data, and on the right, the covariance matrix. So on the diagonal of the covariance matrix, we have the variance of each variable. And we consider that the sum of every variance, so more than 361 and 51, represents the quantity of information contained in the data. So now, let's have a look at what PCA can do for us. So first, I want you to see that using my favorite statistical software, R, like the letter, I can do either the eigen decomposition of the covariance matrix of my dataset data body, or PR comp for principal component on my same data matrix. And here, okay, even if the results are not presented in the same way, on the left here I have eigenvalues, and here I have standard deviations. But if you take the square of the standard deviation, you will have the eigenvalues on the right, my right, your left. Okay. If I have a look at the eigenvectors, I have up to a sign the same that is provided in the rotation object with PR comp. But in PCA, we don't care about the sign of the eigenvectors, okay? Because we just have to interpret proximities between individuals. So whatever they are on the right or on the left of the plot, it will be the same interpretation. So actually, we indeed have the same information when we run a principal component or an eigen decomposition of the covariance matrix of a data set. So what does this mean here? Here, this provides the coefficients of my linear combination or the loadings. It's frequently used, this word. So PC1, my first principal component, is defined by this linear combination. So 0.45 times shoulder girth plus 0.32 times chest girth and so on. And PC2 in this way and so on. So let's go on with this. So the idea with PCA is to be able to work with a centered data. So to center a data set, you compute the mean of every column and you remove this value to every value in the table. Okay? Then on this centered matrix, you apply the loadings so that you collect the coordinates of each individual on the principal component. And what the matrix algebra ensures you is that the variance that you get on this column, so the coordinates of the individuals on PC1, is the greatest variance you can obtain with a linear combination of the initial variables. Okay? And after, in an orthogonal direction, you have the greatest variance and so on. And you can see that if you add these variances, you get the same quantity of information. There is no loss of information as soon as you take the same number of principal components than the initial number of variables. But the idea that was written on one of the texts from Wikipedia is that we can reduce the dimensionality of our data set. The idea is that if you focus on the first two dimensions, you will have roughly 90% of the information on only two dimensions. Okay? So that's the idea we use to produce this kind of plot. So here you have on the top what is called a scree plot. So scree plot, you can display either the variance itself or the percentage of variance explained by each principal component. Okay, so here you can see that with the first two principal components, we have 90% of the information. So that's the deal that PCA proposes. He said, okay, you add 100% of the information in 5 dimensions. Here is my deal. You keep 90% of the information with only 2 dimensions. Is it okay for you or not? If not, okay, you seem to be tough in trading, so I can propose you nearly 7% more in 3 dimensions. So we have to see, depending on each data set, we can choose 2 or 3 dimensions, 2 or 3 principal components to display our graphical outputs. And there, if we consider the coordinates on the principal components, I can display my 20 individuals to see if they look like in the same way, okay, after projecting on the first principal component. What can also be represented is this kind of plot, can be called loading plot. It's just a bar plot considering the loadings, so the coefficient of the linear combination or the eigenvectors. Having a look at these and these, we can interpret, you know, the idea of the beefiness I represented with the 3 dimension before. So here the idea would be to try to interpret to put words on my principal components. It can be also easier representing the variables in this way. So computing correlation between initial variables and the principal component. Here 0.87 is the correlation between shoulder girths and PC1. So I can locate shoulder girths at here 0.81 on the horizontal axis and minus 0.15 here on the vertical axis. In this way, I can start interpreting my principal component, telling that every people I find on the right of my plot has rather high values for every of the variables on the right. If you are on the left, you have probably a lower value of the 5 variables. After, if you consider the vertical axis, you can see that you have shoulder girths and height at the bottom, and waist girths, chest girths, and weight at the top. You can see these, okay, you on the right, you seem to be tough. But you can be tough because you have big muscles and you're tall and you're kind of maybe Flora Manodou, you see? Rather a tough guy. Or if you have there, because of waist girths, and fat and so on, like Homer Simpson, you see? So okay, you're tough, is it because you're athletics or because you're drinking Duff to Mouse, you know? Homer Simpson, okay? Okay, and another graphical output is the biplot. So biplot is the superposition of individual plots and correlation circle plot, so that you can have a picture of your data set. So here, for instance, I have just put some colors, and I hope that everyone can see colors. Okay. So here, for instance, high 7 in yellow there, okay, is on the right, nearly on the top. If we have a look at these, okay, shoulder girls 123, the highest value there, 184 at the height, 86 kilograms. Okay, what else? Someone rather tough. If you have a look at I18, someone rather skinny probably, and so on and so on. I try to highlight some specific body shape. And another aspect to have in mind is that the origin here, at the origin here, we have the average profile. And in this way, you can think about the location of every individual in relation to the average profile. So for instance, I3, so moved away from the origin following shoulder girls. Okay, so high value here in shoulder girls, and also probably the tallest person in this sample. Okay? So you have seen that we come from matrix algebra applied to statistics on a data set to try to have a picture of a data set that is not so easy to understand if we just have a look at the numbers. So just a few words about how to interpret the results, so that correlation and cosine, you have the link between what are variables that are correlated, and the cosine we'll have between the two arrows that represent each variables. It's something I hadn't told before, is that variables are represented as arrows. So you can visualize the angle and think about the cosine as the correlation between the variables that are represented as arrows. Okay, for the individual plot, so we have to interpret the graphical outputs of PCA, but keeping in mind that we are looking at a projection. Okay, so we have to be careful when interpreting visual proximities. So I would like to illustrate this in comics with the only true superhero. Do you know who is the only true superhero of the comics? No? Superman, Spider-Man, Batman? No. Imbattable. Does anyone know Imbattable? Sorry for you. So, Imbattable. I have something to... So, imbattable is a word in French for someone who cannot be defeated. Okay? So why you cannot be defeated in comics? Oh, sorry, you cannot see. So he cannot be defeated because he can jump from one cell to another. Okay? So it's easy for him to catch a thief, even if he can run very fast, because he can jump here, he can be there to be stop him, and so on and so on. So very nice comics. So, and this guy, imbattable, once he meets this young guy. His name is, you can maybe read it, 2D boy. So the boy XY who doesn't care about the Z. So what is the superpower of 2D boy? Let's have a look here. Okay, we think the tree is far behind him, but he's a 2D boy, okay? So here in this way, I could shake the hand of Jérôme and there. Okay, hi Jérôme, you're there. No, okay? So 2D boy. So why am I talking to you about imbattable and 2D boy? It's because sometimes we can think in this way. Okay, I have my two people, I13 and I14. They seem to have the same measurements because they are close on the plot here. But if I have a look at their measurements, they are not so similar. Why? Because let's think that I am projecting my observation on a specific plan. And if I have a look at the third dimension, I13 and I14 are not so close. Okay, just always keep in mind this. Okay, so as a wrap-up for graphical outputs, so we have the script plot to say how many components I have to get to display my graphical outputs. So let's think about a deal. Okay, you have 90% with 2 dimension. Do you want more with the 3rd dimension or not? Individual plots, so natural clusters, outliers, careful with the visual proximities. Think about 2D boy. And variable plot or loading plot. These are the correlation between variables, and it can help to interpret components. So here, for instance, on the toy example, PC1 a kind of beefiness and PC2 a kind of fatness or rotundity, something like that. Okay. Where am I? So, yeah, okay. I can just show you 3 simulated examples just to be sure that PCA is okay for everyone. So if I simulate here 3 data sets with 50 observations and 3 variables. In the first case, on your left, I have 3 variables with no specific link, nearly 0 correlation for the 3 pairs of variables. In the second one, in the middle, I have highly connected V1 and V2. And V3 is not correlated to V1 or V2. And in the first case on your right, the third case on your right, so I have highly connected the 3 variables. Okay? So you can think about each data set as a ball, pizza box, and a tube. Okay? So let's have a look at the result of PCA. In the first case, for the ball, here PCA is trying to catch a specific direction onto my ball would have an oval shaped. Okay, even if in Toulouse we do like oval shaped ball, but here it's a football. So here PCA doesn't know what to do. It provides 3 principal components with roughly 1/3 of the information. If you have a look at the second example, so here we have the loadings, here we have the script plot, and the script plot tells us, okay, it seems that you can gather nearly all of your information in 2 dimensions, and the third one can be roughly removed. And here on the correlation circle plot, we have a very sharp angle between V1 and V2, that's the visualization of the high correlation between V1 and V2. And in the third one, with the tube, okay, I have nearly all the information in only 1 dimension, okay, because of the high correlation I have between my 3 variables. Okay, so all I've talked about so far is something you will probably learn in lecture, not so far if you follow lecture in statistics. Now we are going to make a step in direction to research, because now we're going to talk about extension to integration problems. So integration, data integration. So the idea is that, okay, so here now I hope that you have understood that I can run a multivariate unsupervised method dealing with one numerical data set running principal component analysis. As soon as I will have information on my data set, I can consider this kind of method. So now I have one numerical data set and one categorical variable. So now I can extend my PCA to a linear discriminant analysis or to other methods such as projection to latent structure in discriminant analysis. If I have several numerical data sets, I can go through a generalized canonical correlation analysis or a multi-block projection to latent structure method. Now, the next step is I add this kind of stuff here. So I have now a multi-block supervised. So supervised is when I want to focus on a categorical variable. Okay? Okay, so multi-block projection to the latent structure discriminant analysis. And another aspect that I can deal also with multi-group analysis. So multi-group analysis is the same as above, but with the samples. So samples are individuals, observations, divided in predefined groups. Okay, divided in predefined groups. In is badly placed. Okay, so either batch or study and so on. It's very useful when you want to run what is called meta-analysis. So you gather analysis from several data sets everywhere in the world and you want to run a meta-analysis. It can be useful in this way. So the idea here is to extend the principle of PCA. So PCA, I hope you have in mind that we do something to maximize the variance. Okay? And I build principal component. So the idea behind the extension around to integration problem is that now I can maximize not only a variance but also a covariance. It can be done with metric algebra or with specific algorithms, but the idea remains the same. I want to build linear combination of my initial variables, optimizing a specific criterion. PLSDA is here because we use this trick is that I can convert a categorical variable into a numerical one built with dummy matrix. Okay, it can be a kind of cheating, but it works. So actually, we do this kind of stuff in data analysis. And after, if I have several blocks, so in a multi-block analysis, the principle is the same, except that the criterion to optimize is more complicated. Okay, it's just potentially weighted sum of covariance, pairized covariance between each data set. Okay. And another aspect to extend to integration problem is that we can also take into account a sparsity using some rather classical aspects of Lasso penalty so that we can select the most relevant features in a data set. And here is the visualization or schematic view of what can be done with a sparse PCA. But we can run a sparse PLS, a sparse PLSDA, sparse multi-block PLSDA, and so on. Okay? Are you okay? Everything is fine? So let's go back on track. Do you remember my question? Real world question. How is global warming affecting plant growth? So now we have a toolbox with, I can have a look, PCA, PLS, multi-block PLS, multi-block PLSDA. Okay. So let's see how I can use these tools to try to answer this kind of question. So I'm going to talk to you about a project I've been working on with colleagues from the university. So that went to the Pyrenees to collect plants on the ground. They gather seeds and grow them in controlled conditions. And the idea is that to control hygrometry, temperature, and so on, light, and so on. And use two different temperatures, either 15 or 22 degrees Celsius. Once they have grown, they collect different parts of the plant, so stem, leaf, rosette, and so on. After, they make a kind of soup with this, and they put it in a very expensive material such as DNA sequencing, mass spectrometry, and so on. And once they have done that, they generate very large data sets. Here we just only an extract. For each of the biological sample, they can gather thousands of features from DNA, from the genes, from the RNA, from the proteins, and so on. So, if you remember the quote from the book by Searle, so biologists need to, they need skills beyond their general experience. So they need someone to help them in analyzing this kind of data. So the data looks like that. Actually, we had put the data in an R package. So at this web page, you can gather all the data generated in this project. So we have 60 samples of Arabidopsis thaliana for 5 ecotypes grown at 2 temperatures with 2 organs, stem and rosette, 3 replicates, and we have 4 tables, 4 data sets corresponding to proteins, proteomics, more than 20,000 features, 400 transcriptomics corresponding to RNA gene expression or RNA production. Sugar can be also called metabolomics, and phenomics, just measurements on the leaf, on the rosette, on the stem, and so on. So, in this context, one specific question was, can we determine a multi-omic signature to classify ecotypes on the basis of floral stem data? Okay, let's try to translate this into a statistician vocabulary. So first, multi-omics. Multi-omics means that we consider all the data sets. So proteomics, transcriptomics, metabolomics, phenomics. So for us, it's a multi-block analysis. A signature. Signature for biologists is that I want to select the most relevant variables inside each data set. So I have to use a sparsity. To classify ecotypes. Classify ecotypes means I have to use a supervised method. So I have to run a discriminant analysis. So now I have all the pieces of my puzzle. If I consider that I have to filter my data on the organ equals stem. Okay? So now I know that I have to run a multi-block, a sparse multi-block discriminant analysis. And this method was published, it was in 2019. Okay, so we are talking about actual research, okay? Current research. Here I just put the formula that looks like what I've told you about a weighted sum of pairwise covariance. And the idea after is not to provide this to the biologist, take them, okay, you just have to apply this and good luck. No, so I have to run this and propose this kind of graphical outputs that are mainly based on the same as PCAs, but with other specific plots. Here is the same as individual plot for PCA, but as we have 4 blocks, I can have 4 individual plots. Here I have a correlation circle plots, but with 4 sources of variables. Here, I do not like this, but it looks fancy, so biologists can like it. Okay, but for me, it's highly difficult. It's just to display correlation between variables from each data set. And here is something you have on the poster of to announce this conference or this lecture. Okay, this kind of plot where you can link the biological samples to the variables that have been selected. And here the color band indicates the ecotypes, the 5 ecotypes of the study. And here you have a color band indicating where does the variable, the feature selected come from. So from which data set it comes from. So here, okay, it's something that is difficult to interpret, but that contains the answer to the questions. Can we determine a multi-omic signature to classify ecotypes on the basis of flower stem data? Okay. Okay, so yeah, okay, I think I can finish in 5 or 10 minutes. So this Wallomics project lead to several publications. So some in plant science. Okay, I have been associated, but the main message was to the biological community interested in plant science. Here it's Frontiers in Plant Science, another one. Here in a journal called Cells. And another one, I do prefer this one because what we have published in this article in 2020, I think, is the framework where we have described, okay, here are the data we had collected to study our problem in plant science. And here is a network we have built to analyze and to have a comprehensive overview of what was inside the data set we collected. Okay, and here I have just put to show you the different roles in the work, in the publication, coming from the PhD student, from the intern in applied mathematics, to the researcher and the PhD supervisors, and the guy, a statistician, contributed to this. So to finish, I would like you to show other problems in the real world where math can help. So once again, based on a real world problem. So does wearing dental braces for 18 months really work? It's important. Okay. So for a shining smile like this one, you must go through this. But this for I don't know the word for orthodontist, but for okay, for someone who puts the teeth in the nice way. Okay, I cannot remember. Okay, so this guy, Dr. Maxime Rothenberg, see teeth and so on, but as a mathematician, don't you see something different? Don't you see? Yeah, and actually it was the thesis of Dr. Maxime Rothenberg, so modeling the dental arc with a 4-degree polynomial without odd degree terms for actual symmetry and so on. So the idea is that once you had wear your dental braces, so those your teeth are better on the mathematical model of ideal dental arc. So it can be useful for that. Math helps to understand cheese ripening. Nice. What are the microbiological mechanisms involved in cheese ripening process? So actually, that's a project, a European project I'm involved in now actually. And it's EMUS project, so complex microbial ecosystems, multi-scale modeling, mechanistic and data-driven approaches integration. So seriously, we are 10 European partners in 8 countries in Europe gathered to better understand the cheese ripening process. Okay, if the same idea is behind what I've shown about plants. Okay, if you do like cheese, maybe you don't want to know what is on the surface of a cheese, okay? But if you look with a microscope, not sure you'll want to eat cheese again, but there are many things that can be studied, so microbial communities that compete to build a cheese and so on. So it's very interesting. And in this context, so there are, of course, I think there are, yes, 15 PhD students in this project. One of them is there, I co-supervise him, is in the next building. And his work consists in studying kernel approaches for the integration of biological data from heterogeneous sources. So just briefly, the idea is that, okay, how long does it take to go from point A to point B? Okay, what is the distance between the 2 points? Okay, okay, just measure that. But I can also go in this way. Okay, so it depends on the way you measure the distance. Another illustration is this map. Okay, in green you have the well-known map of France. And in red, you have another map of France, but it's not kilometers between cities. It's the duration of a trip by train from Paris. And Toulouse is rather far from Paris if you take the train. It's not actual, it's in 2015, but roughly the same now. And you can see that Bordeaux is very close to Paris because of the high-speed train. Okay, so the idea is to study this, to develop this kind of approach through kernel approaches, and to see that, okay, how could I divide my red squares from my green circles with a linear shape? How could I do that? Not so easy. But it can be done if you project. Once again, it's about a projection. You project on a specific shape, and after it's easy to cut in a linear way to distinguish your red square to the green circles. Okay, so the shining smile, the cheese, and I think that someone, at least one, already heard me about math helps to optimize performance in sport. So the idea is that if you know maybe the movie Moneyball, okay, it's based on a book, the same name, Moneyball, and Lisa Simpson, when she wants to be very serious, she has stats, of course, EIP plus 1 equals 0, F equals MA, and Moneyball. So it's not, it's a very good, very famous book. And here, so I've just learned this morning that this guy passed away in the next few days. So you know Brooklyn Nine-Nine, the series? Okay, you should have a look at this, it's rather nice. I think I have, up. Voilà, that's my favorite. So the statistical analysis is so beautiful. So I would say in memoriam to this guy, that's okay. I've learned this morning that he passed away. So I'm a bit sad because it can be, he feels something in front of a statistical analysis. So it's something amazing. So yes, for optimize performance in sports, so to hide new players, to prevent injury, to model collective behavior, to identify optimal strategies and so on. And here, okay, so quand les maths se mêlent de sport, so when math are mixing with sport, okay, so there is something on the Journal of CNRS. So you can see many articles on this topic. And just to finish, I would like to show you examples in other domains, so in which I've contributed. Those are publications that I'm a co-author of these publications. So in a journal of cerebral science, so to study the differences in breed protein digestibility, so just to find varieties of wheat that are easier to digest. Journal of Voice, I've been working with a voice therapist to help what is called, yes, sports and fitness instructor to not to have a problem with the voice, because all along the day you're going to say, okay, move to the left, to the right, to the left, to the right, and so on. Yeah, okay, with me, okay. All along the day, at the end, it's difficult for their voice. Probably the same for teachers that speak all along the day frequently. This American Journal of Clinical Nutrition, it's a study about obese people trying to understand how they can become, how they can develop disease or not. Okay, cancers, of course, radar research, so how to understand microbial communities that can help to depollute waters, in psychiatry and in computer science, or here with studying the RNA profiling development or a specific shark. Okay. Specific sharks, they study the head of the shark to see what happens in the head, how they develop. Okay. So that's the end for me, so I think we have time for questions, so if you have, I'll be happy to try to answer them. Thank you for your attention.
Thank you very much. Any questions or comments?
No, everything was very clear, very interesting. So if you don't ask me any question, I will ask you a question. Okay, good. No, seriously, don't you have any question? No, everything was clear. What was your favorite part? What was your the part where you hadn't understand anything? I can have a question for you if you don't have for me. No?
Actually, I have a good question. So, you showed that very, very small data sets. But if you take very large data sets, you will need a lot of computational power, or can you perform this kind of analysis on a laptop?
Good question. No, actually, I have never needed something different than my laptop. So even with thousands of features in a data set of columns, okay, we can deal with this kind of laptop. Because when it's linear algebra, it's very efficient, so it's very fast. Okay, sometimes I can spend a few minutes, but I never needed high performance computing, for instance.
And are you developing the algorithm related to linear algebra yourself, or are you using pre-installed software?
It can be both. So So if something is already developed, I will not develop again eigen decomposition, something like that. But if the idea of building on our package is that you rely on existing packages, and okay, I depend from this existing package, and I have added these functionalities. So it can be a mix between.
How do you get to work with all these different people, and how do I know aware of all these different projects? Are people coming to you and say, okay, I have brought this topic, can you help me? Or how does it work, the collaboration? How does it start?
So I would say frequently it starts in a training event, because I am used to train biologists to statistics, to mathematics and statistics. And at the end, I say, okay, here is my card. If you need, just give me a call. And okay, oh, I attended your training workshop, and it was very nice. I have to believe it. And maybe you could help me in this project. Okay, yeah, let's discuss, and so on. And I would say that behind all these publications, I think that there is someone I have trained in statistics, actually. Yeah. And after what is called in French, bouche à oreille, once I have worked with someone, we say, okay, maybe you could send an email to this guy. Maybe he can help you. And so. Yeah.
Are they all academic projects or also private companies? Or?
Mainly academic projects, but not exclusively. If you have seen here, Christian Hansen and Nizor are industrial partners of the European project, and also French company for milk industry interested in these developments. Yeah.
Last chance. If not, we can have a coffee break and ask questions during the coffee break. Thank you very much.
Thank you.
Okay, so we are going to resume this masterclass session with Jérôme Fehrenbach, who will speak about modeling and optimization to remove strips in an image.
Stripes.
Stripes.
Okay, thank you, Stéphane. Okay, so I will present some work that was done with my colleague Pierre Weiss. It's not a very recent work, but I think it is the most impressive visually, because we worked on image processing. So here again, like the talk of Sébastien earlier in this afternoon, it is mathematics applied to real life, and the application is to image processing. So first, I will present the motivation that started when we met some biologists who had some specific microscope images. Then I will present what is quite classical Bayesian parameter estimation. So I will mostly focus on this, which is a general tool. So even if you are in another branch of mathematics, anyway, this is quite important to follow. And then I will present more rapidly the models that we propose. So it is called VSNR, a variational stationary noise removal, and I will present the results that we obtained on the images. So first, the motivation. The biologists that we met, they had some special microscope, which is called a light sheet microscope. So with some optical device, they turn a laser beam into a laser plane. So the plane is here, and it illuminates the sample, and you have a microscope, and you look at your sample. So it is that kind of image that you have. So maybe it's not very easy to see, but since the light comes from one direction and it may be absorbed by some parts of the tissue, so you can see that the image is impaired by some stripes here. Everybody would think that here it is some thing, maybe a cell or a cell nuclei. Here also you can imagine one, but you see that with all these stripes, it is because you have a brain that you know that there is something, but it is very degraded. The image is degraded by stripes. So I think I have some other examples. So here you can see the things, but you can also note that there are some horizontal stripes that affect the image. Here is still another example. Always the light comes from the left and it gives horizontal stripes. Even in 3D, so for instance, the biologists, if they want to quantify some things, they want to detect there are some nuclei that are quite round and some that are quite fuzzy, and when it is fuzzy, it is that the cell is dividing, so it is important for them to make the difference. And so they have to make a segmentation to detect the nuclei, and after to know if it is a nuclei of a standard cell or the nuclei of a cell that is dividing. And if you make some detection because of the stripes, here the detection is bad. You detect something, but it is not very good. Here also it is not so good, and in these places it is also not very perfect. So that was the question that was asked by the biologist, and in all the image processing tools that existed, they could not fix this stripes problem. So we decided to work on this. Oh, okay. So here is, I have other images. This is from another modality. It is atomic force microscopy. So you have your sample and you have an arm that moves along the sample. And since it moves in one direction, after it comes back, it takes the next line. After it comes back, it takes the next line. And that's why you have some like horizontal defects. And here is another image in atomic force microscopy. So your brain can see what there is, but if you want to make some automatic quantification from this image, it is very difficult. So, well, okay, I wanted to change this. Okay, so now I will come to Bayesian parameter estimation. So I will switch to the blackboard. So everybody knows Bayes formula. It is about what are my notation? Yes. Conditional probability. The probability of A knowing B is the probability of B knowing A multiplied by P of A divided by P of B. Is that right? Yes. Okay. So how can this help to solve things? So I will first. So images, you are in a large dimension because you have pixels. Typically, when your image is 1,000 pixels by 1,000 pixels, so you have 1 million unknowns. So you live in a very high dimension. So first, I will take one-dimensional examples to know how we can apply. Yes. Oh, this is Bayesian. So I went to the grocery store, and I bought some clementines from Corsica. They are very, very good. So I have a certain number of clementines. And I asked to the seller, what is the weight of one clementine? And he said, well, it's not a perfect weight. So it's about 100 grams plus or minus 20 grams. So the weight of one clementine, so I call it, okay. So this is my prior, A, the weight, and it is distributed following a Gaussian law. So it is a Gaussian law with expectation 100, but standard deviation of 20 grams. Okay? And, well, this is not, well, this is the prior information that I have, so the information that my seller told me. So I want to know what is the weight of this clementine. How can I do? Well, I brought something else. I have a scale. So I put it on the scale, but my scale is not very, you see, it is not very precise. So it says, okay, it says 90 grams. When I look at what my scale says, it says 90 grams, and the uncertainty is 10 grams. So I call it B. Okay. What I measure for this particular clementine is 90 grams with a standard deviation of 10 grams. So how can I have some more information? So I will use Bayes' formula here. So the A, sorry. So I will write it another way. So the event A is the weight of my clementine is X, and I know that the probability of A, it follows a normal law with expectation 100 grams and standard deviation 20 grams. And what is B? My event B is measure on a scale. I read X prime. Okay, so this is my event B. And what do I want to know? I know that I measured X prime. So, sorry, I want to know what is the probability of my weight being X, knowing that I read X prime on the scale. Okay, so how do you write this? So this is a scale. The scale measurement, I know that my scale has a Gaussian error with standard deviation 10. So if the probability of B of reading X prime knowing A, knowing that the true weight is X, so it is proportional. With the Gaussian, you have some factor in front. I ignore this factor. So it is exponential minus X prime squared divided by 2 multiplied by the standard deviation of your scale to the square. Okay? So this is the probability of B knowing A. And so what do I know in this formula? Probability of B knowing A, this comes from the error law on my scale. The probability of A, this is my prior. So this is what we call the prior. And so the probability of B, in fact, since you make one measure, you read 90 grams. So B, the probability of B, it is it is not a stochastic event. You read 90 grams. So what is behind the Bayesian parameter estimation? So I want to find this, and maybe I want to find what is the weight of my clementine that is the most probable. So what do I want to find? I want to find X. So, okay, so this is my prior, so the posterior law. I can write it, probability of B knowing A up to a constant factor, it is a product of this and this. And this is a constant, and you have also some constant factor. So it is exponential minus X minus 100 squared minus X minus 90 squared divided by 2 sigma prime squared. Okay? Sorry. Yes, it is. Okay. So the question is, I want to find the maximum a posteriori estimator. So this is the true probability of A knowing B. Is find X that maximizes this probability, that maximizes P of A knowing B. If you want to say it in another way, you want to find what is the most probable weight for your clementine, for this precise clementine. So I want to maximize this quantity, and this is equivalent to X to minimize minus log of this probability. And why do I take minus log? Because you know I have some exponential, so I want to find the minimum X. I want to find X that minimizes X, so it is only 1D, X minus 100 squared divided by 2 sigma squared plus X minus 90 squared divided by 2 sigma prime squared. Okay, so this is Bayesian This is Bayesian parameter estimation. I have some prior, and I also have some measurement. And I want to combine both of them to find what is the most probable parameter x. Okay, and in this case, it is very easy because it is quadratic. Okay. So you have a quadratic function to minimize. So you have directly the solution. Okay. What? So this is an example in 1D. If you work in general dimension, if you have some Gaussian prior here with a given covariance matrix, so and you have also for your measurement noise a Gaussian noise, then you will end up here with two quadratic terms involving the covariance matrix of your prior and the covariance matrix of your noise, and this can be solved. Well, if you have some you want to minimize some quadratic function, you have only to solve one linear problem. Okay, you cancel the derivative, and to cancel the derivative, it is a linear equation. So this is it. And what can we? In fact, this can be used for image processing. So I will first present what are the standard image, what are the standard use of this in image processing, and after I will show what we added in our to solve our stripes problem. So okay, I cannot write on this second board, so I will erase this one. Classical maximum a posteriori estimator in signal and image processing. So what kind of prior would you have? You want to say my image you want to look for a smooth image. Okay, you can say, well, if it is really an image, my image is smooth. So what does that? You see in the functional that you have to minimize, this term would be like 1/2 of. Okay, I write it here. The term would be in the norm of the gradient of your image. Okay, U is your image. And what are your measurement error? This is a noise model. you can say that you have some Gaussian noise that is pixel-wise independent. So, which means that every pixel is affected by some noise that is drawn from a random. What would be the term corresponding? It would be like 1/2 of your image minus your observation. Well, you remember I had 1 divided by. I have some covariance, some variance here. So if you want to look for a smooth image with a Gaussian noise model, you must minimize the sum of these two quantities. So you want to minimize with respect to u 1 divided by 2 sigma 2 norm of grad u plus 1 divided by 2 sigma prime squared times u minus your observation. In fact, you have two constants here. You can multiply by 1 constant, and in the end, you finish with only 1 parameter. So here you have 1/2. And here you have alpha multiplied by 1/2. Okay, this would give you the same minimum, and this is the regularization parameter. Because in practice, nobody will come and tell you, well, the variance of the noise has this value. Nobody will come and tell you, well, the probability of your image depends on the norm of its gradient with some variance coefficient that has this value. So in practice, you play. In fact, it is the quotient of these two quantities that gives you this regularization parameter. And if the noise is very strong, it should be taken high. And if the noise is small, it should be taken small. But anyway, to find the value, you have to check by hand. So this is the first. The first model, we call it H1L2 model, because you have a H1 prior on a norm of the gradient and L2 for the noise. And look, it is some quadratic function, and to minimize this function, you just have to solve a linear equation. So I will show you. I will show you some result. So this is, I will show you some 1D result. This is signal, it is not an image, it is a signal. Well, you can imagine that it is affected by noise. Okay, it is some nice smooth signal affected by noise. So you can plot your signal, and then you only solve, well, my regularization parameter, I call it lambda. Okay, sorry, it's not alpha. So K is the Laplacian matrix, because it is a gradient transpose gradient. If you want to minimize, you have to take Okay, and what does it give? For instance, with lambda equal 1, with lambda equal 1, you see it is close to the black curve, but it is more smooth. Well, but it is not smooth enough. So I will take lambda equal 10 and I plot it in red. Okay, lambda equal 10, it starts being better smooth, but still has some small oscillation. So I take a larger lambda. I plot it in green. So now in green it is very smooth, but you see that in fact what is not very nice here, in fact there is a transition between this part that is going up, here there is a sharp transition, and this part is going up. And you see that doing this, using this approach, I have some smooth transition. Of course, because my prior is to say that my signal is smooth. So of course I recover something smooth. But if you think of especially images, it is not smooth. It is usually piecewise smooth. You have one part that is black, you have a boundary, you have one part that is gray, you have a boundary, you have one part that is white. So this leads to another prior. Instead of saying my image is smooth, I say my image is piecewise smooth. And what how can we translate this? It is some prior that would penalize the total variation of u, which is the integral of gradient u. Well, at least if u is smooth. So you see instead, okay, so this is the integral of gradient u squared. So it is not the same thing. I penalize the norm of gradient u and not the square of norm of gradient u. And if I use the same noise prior, so I obtain what is called the TV L2 model. So I want to minimize some regularization multiplied by TV of u plus this L2 term. Okay, so I change my prior. And now you see that it is, I will not get the solution as easily because here it's two quadratic functions I can minimize by just one linear solve. Here it is a complex optimization. It is convex. It is convex optimization because this is convex, but it is non-smooth convex optimization. Okay, so I will not get into the detail of this, but because we should need more than 1 hour for this. So this is non-smooth to solve. It is a non-smooth convex optimization problem in large dimension because here this signal, the size of this signal is, I don't know, maybe a few tenths or a few hundred, but for an image you have the same, but the size of the signal is a few thousands or 1 million. So what is nice is that it is a convex function, so you are sure that there is only one minimizer. So you can be confident if you have a good minimization procedure, you can minimize this. So this is what I did for this, and I use some library for this, and I have some okay. The parameter here is, I call it lambda. And now the denoised signal is a red signal. So you see that it is piecewise smooth, but the transition here is still good. And now I will increase, but anyway, I don't really like this red signal. It has too many oscillations. So if it has too many oscillations, I need to make alpha become bigger, to make to give more importance to this term. So I change with some larger alpha. And now you see you have your very nice signal. In fact, for this TV, always when you have TV terms, in fact, you have what is called the staircase effect. So now, anyway, the thing that I have is that my signal is really piecewise smooth. So this is classical things. I will show you just for the pleasure of the eye. Okay, I have one image. Now, I don't remember what is the size of the image. Okay, it is 512 by 512. So you have 25,000 pixels in your image. So the original image is here. I put it large. I add some noise. So this is what I call my observed image. Okay, and when I denoise it using, so it is here, using the TVL2 model, you see that it is okay. I show you with the original. You see there are some. Well, there are some differences, of course, if you look here, but it is very close. And this is because we use the TVL2 model. If I don't have it, but the H1L2 model, it would make some blur in the interface. Okay. Is that okay? There are some questions? Yes?
So when you want to find a new image, you want to find the U, but you made before some example for the choose of alpha, and in the case of the image, what are you considering to choose an alpha and alpha?
Well, yes, I can answer. Well, you will see the result with a larger alpha. I multiply by 10. So, well, in fact, there is no a priori choice for alpha. If you know your device, if you know what is the typical noise of your device and what is the typical type of image that you want, that is, what is the typical value of this term and what is the typical value of this term, then a typical value of alpha would be the same. But if you work for the first time on some project, then you should try a lot of values. Okay, so now if you increase alpha, I compare with the other one. So this is with alpha equals 0.04. If you take a larger alpha, you have it is much more piecewise constant. So you can make some nice. You can play nicely with the images. Of course, it is not the original image, but it is quite nice to look at. Well, so you. Other questions? Okay, so now okay, I had prepared this in case the code would not work. So this is all we had seen. So now I will come to our contribution. What we propose, remember these images that were impaired by stripes. So now the problem is the noise model is no more valid, because if you have a stripe, of course your stripe it is the definition of your stripe. It affects several neighboring pixels. So you cannot use the fact that your noise is pixel-wise independent. So this is what we have to modify. And there is something we use. It is the fact that all the stripes have the same direction. You see, you remember in the images that I showed, all the stripes have the same direction. Of course, if you have no stripes anyway, you cannot hope to do anything. But if you use the fact that all the stripes are in the same direction, you can hope to make something. So what is the proposed noise model? It is the fact that my noise is, I have some, for instance, my image is here. I have one stripe here that I call psi, and it is psi, but in several locations. So how do you move some pattern in different places of your image? You use convolution. So it is my pattern psi convolved with, how do I call it? Yes, convolved with some lambda, and lambda is ideally it is a sum of Dirac. So if I have lambda is what, and now lambda will be, it will be pixel-wise independent. So for the lambda will be here, 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 and here. And so if I convolve my pattern with the lambda, I will have different stripes in these different locations. So that is the idea. In fact, we consider different elementary stripes, and in fact, with only two stripes, it would be enough. If you take for psi I, if you take only one pixel, then it will give you some noise, some pixel-wise independent noise, and we want to add the possibility of having pixel-wise independent noise just for measurement noise. So okay, we can think that we have one or two basic patterns that are placed all in different places in our image using this convolution. And after what? So since this is our bio. Okay, so I will write finally the variational problem that we have. So in fact, So we want to minimize. So we want to find the minimizer. In fact, what we want to find is not the image U, but it is the noise, it is the lambda here. So I want to minimize, to find the minimizer lambda I, such that this is my observation, and I subtract the noise. So Ci convolve with lambda I. So if I take my observation and I subtract the noise, then I should have some nice image, some say piecewise constant image. I want this to be small, and what I want for the lambda I want to have values only in some places, and this can be ensured by some L1, well, by some penalization. And the penalization of lambda I that we used is some L1 penalization. So we started this. In fact, we don't exactly use the TV. Okay, I wanted to cheat for you, but in fact, we take, okay. In fact, we don't use exactly the TV norm, which should be non-smooth here. We prefer to use a smooth version. We prefer to use a smooth version with a size epsilon. It would okay. Maybe what time is it? Okay, I don't write the details, but well, you can have. So now, okay, I call it TV epsilon. It is just a smooth version of TV, and you see that this is a convex problem, but it is well, this part is smooth, but it is not quadratic. Okay, and this part is not smooth. And in fact, we worked with this. We work with this model. So every time the minimization took a long time, maybe for 1 minute for one image. And after we discovered that it was if we take the squared L2 norm, it gives the same results. And this comes because of the law of large number. If you make a convolution of your pattern which has a large number of pixels, in fact you have essentially the same minimizers if you take a quadratic term. And on the numerical point of view, it is faster to have to minimize a convex function, which is an ugly function, well, not so ugly, smooth, convex, plus a quadratic. This is faster than smooth convex plus convex. So I will show you for the noise model. So for the noise model that I propose here, so these are realization of stationary noise of that kind, well, with only one elementary pattern, but it looks like stripes. Okay. for the minimization problem, to minimize this function, we use the state-of-the-art algorithm in the literature by Chambol and Pock, and in fact we minimize f of, it is in which you have two convex functions, and for one you have a linear operator. You minimize f of a lambda plus g of lambda, and where f and g, you can easily compute the proximal operator, but what is not very nice, it is this linear operator here, and well, there is an algorithm that you can apply, and you see this linear operator, it corresponds to the convolution here. Well, so I will not get into detail, and just show you the results that it gives. So this is the original image, the observed image, and this is what our algorithm gives when you remove the stripe. So I don't know if you can see well, but all that your brain would imagine is done by the algorithm. Okay, so I show you on other examples. So this is another original image, and this is denoised. So the original and denoised. So you see most of the stripes have disappeared. Another example, so here is the original image, and here it is denoised. So you see most of the stripes have disappeared, and if you want to isolate the cells or so on, you can do it. So even for the 3D reconstruction, so this is the original image, this is the denoised one. So this was the original segmentation, and this is the segmentation after denoising. And now you can see the biologists are very happy with all these cell nuclei. They can compute the volume and so on. For the atomic force images also, you see it. Well, this may not be exactly the perfect one, and the last one, it is this one. So if you have this and you describe, you have this. So now you can do what you want for automatic processing of this image once that the stripes have disappeared. Okay, so this is the end. So to summarize, what are the take-home messages is using some Bayesian parameter estimation and the maximum likelihood, it gives you a variational formulation to reconstruct well, signals, images, or whatever you want. And if you use adequate modeling, you can solve real life problems. And well, I did not take, because I think the audience is quite generalist, I did not get much into convex optimization, but you can do really nice things in larger dimension. So I did not note the size of the image, but it is several thousands. Maybe not all are 1,000 pixels by 1,000 pixels, so it could be 1 million unknowns, but maybe it is like hundreds of thousands of unknowns. So every time you solve a minimization problem in very large dimension. But what is nice is that you have a convex function, so you can do things. So that's it.
Yeah, so at some point you said that the psi you use in the sun.
Yes.
Yeah, represents some stripe that you want to move in. So in the choose of the stripe, you use the fact that they come from the side and then.
Here, well,
What are the psi?
Here, just the psi is one horizontal stripe, and in fact it is quite robust to the choice of the psi, but typically, if you have horizontal stripes, you have
So this stripe has some sort of thickness.
yeah, yes, maybe no, 1 or 2 pixels, not so much. And plus, since anyway we want to allow some Gaussian noise, so I have the Dirac. I mean, only 1 pixel is 1, the other pixels are 0. So typically, this is what we use. And for the images with the vertical stripes, you only take 1 vertical, but in fact, it is quite well. My colleagues spent a lot of time to find the parameters, but in the end, I don't know if you take between 5 and 10 pixels, and here between I don't know, like 2 pixels, or well, I don't know. I don't have in mind the parameters that were used, but it was.
How do you know where to move your original stripe? How do you find the Dirac?
No, but I don't move it. It is the convolution that moves it, and the optimal lambda is found by optimization. So I give the shape of the stripe, but the precise location will be determined by this variational problem.
So then it would work for other shapes than just stripes, right? Could you use the same method for other shapes? I mean, if all the noise everywhere has the same shape, but it's not.
Yes. Okay, if you want to see it, I have it somewhere. I have the paper somewhere. Yes. Okay. So you see, these are stripes. You have some orientation, some lengths. So these are the kind of stationary noise that we have. So I am afraid that we have the ugly image. Okay, so we have this ugly image. But here, for this image, so it is a baboon image, and we add this noise plus this noise, that are both stationary noise obtained with, I don't remember what is the pattern of this noise. This is the stripes patterns. Okay, so we have two filters, psi1 and psi2. So I think we have also some other, no, okay, so that's all. But you see that with different components of noise, you can recover this and it is quite impressive that the algorithm gives such good results. We were impressed also. Yes.
Yeah, I was imagining that removing the stripe will use the fact that you use convolution to make disappear something, but in this case this will not work. It's not work using convolution all the images.
Well, yes. In this case, it works very well. Of course, if you have two cells and the separation between the two cells is exactly of the shape of your stripe, then maybe you would add some false information, but. Yes.
Does this technique work for non-stationary noise?
No, because stationary means it comes from a convolution.
Yeah, okay, so really
so
the perturbation is the same, same shape at least.
Yes.
If you have different directions of stripes, it doesn't work anymore.
Well, if you have only two directions, you should give two elementary patterns. But if you have any possible directions, then you should think of something else. Other questions? Yes?
So, if your pattern is stripes, the orientation of the stripes is moving slightly. Is it something that you would be able to detect?
Slightly, how much?
Slightly in the sense
Well-
that maybe the stripes are not perfectly horizontal, so it could be bent like this or it could be bent like that.
I don't know. If you take some oval pattern, in fact, our pattern, it is called the Gabor function. It is some anisotropic exponential plus oscillations, but the oscillations are killed very fast. And well, if you take, so your pattern, it is not like a rectangle, but it has some it decreases close to the border. So maybe you can hope to solve this, but well, you should try.
Other questions on this? If not, we can thank Jérôme again.
Okay, thank you.
And a small gift for